Hey guys, my name is McKenna Stoker, and today I'm going to be showing you about the evolution of HIV. So first I wanted to go over some HIV statistics because this is what got me interested in the idea initially. But today in the world there are according to 2005 statistics, there's 33.4 million people infected with HIV. And 28 million people have died from HIV since 1981. And there's approximately 14,000 new infections every year. No, every day. My bad. That seemed a little too high for me, but that's what my paper says. So I'm going to talk about, first of all, how we figured out the origin of HIV. So there was a virologist at the Tulane National Primate Research Center named Preston Marks, and him and some of his research realized that a lot of the laboratory primates suffered from a disease called simian immunodeficiency virus, which identically matched the HIV virus that we know today. So Marks and his team wanted to know more about HIV, so they decided to go to the home country of some of the SIV-infected primates in their lab and ended up going to Sierra Leone. While there, his team wanted to see if HIV occurred in places where primates suffered from SIV. They surveyed 10,000 people who worked in close contact with monkeys, but they found only 2 cases of actual HIV within the population. Marks and his team was confused because these numbers were so low when every monkey, chimpanzee, and what old world ape in the area had been infected with SIV. So they were wondering why this was happening, and Marks and his team found that in Africa, the SIV virus has been prominent for at least 1 million years, which means the virus had time to adapt and specialize for each species of monkeys. A theory for why it's not as prevalent in humans is because it has not been in humans as long, so it has not had the same amount of time to adapt and specialize like SIV has. So to determine how long HIV has been in humans, a team of researchers named Betty Korber, James Thieler, and Stephen Walensky used a blood sample from the Republic of Congo taken in 1959 to see if the SIV virus had jumped over to humans yet, and they actually found that the blood was infected with a strain of HIV called ZR59. And they estimated that this strain of HIV made the jump to humans in approximately 1930s, which means that HIV has been able to infect humans for less than 100 years, while SIV has been able to infect for 1,000. So now we're going to talk about the initial infection from HIV. So it's important to understand that there's been multiple initial infections with HIV. It's theorized that there's actually about 10 times that it's jumped between humans and primates. But overall, the initial infection had to have taken place when a cutaneous or mucous membrane was exposed to infected ape blood or bodily fluids. So the most likely way this happened was that a bushmeat hunter, which is when hunters go out and hunt indigenous animals to eat, was injured, and his injury came in contact with the infected blood of a primate. So now we're going to move on and we're going to talk about the actual strains of HIV. So there's 2 strains. There's HIV-1 and HIV-2. So HIV consists of 4 subtypes, M, N, O, and P. And these were caused by human to ape cross-species transmissions in West Central Africa. As you can see in this map, we have some arrows showing how they moved. Group M and N are closely related, and they're thought to come from southern Cameroon, and they are also thought to originate from chimpanzees. Group P and O are closely related from West Central Africa. There's not enough strains of these to actually be able to pinpoint the exact location where these transfers happened. But it's theorized that Group P came from gorillas. But Group O remains a mystery because there's no ape virus that closely matches HIV-1 Group O enough to say that there's a comparison. So groups M and O likely emerged at the beginning of the 20th century, while N and P emerged more recently, but they are still so rare that we can't make any definite conclusions. Meanwhile, HIV-2 was mainly restricted to Guinea-Bissau and Senegal. HIV-2 is closely related to Sudaimangabes, a primate living in West Africa. HIV has multiple subgroups, A through H. A and B are the only ones that have actually been able to spread to humans in a recognizable degree to where we can actually study them. And the other ones have had such a limited spread and only been identified in a few individuals that they can't really offer us any information that we need to make definite conclusions. Okay, so now we're going to move on and we're going to learn about how HIV attacks the host. And first we're going to do a little bit of backstory on how our immune system protects ourselves so we can understand just how it affects. So in a normal human being, we have CD4 positive helper T cells and CD8 positive cytotoxic T cells. And from now on, I'm going to refer to them as helper T cells and cytotoxic T cells, respectively. So helper T cells identify epitopes on bacteria, toxins, or virally infected cells. And once they identify a specific epitope, on a macrophage, the cell begins to divide and secrete proteins that activate and promote components of the immune system. This immune system reaction helps trigger the cytotoxic T cells, which hunt down these cells containing these epitopes. And then they bind to the epitope and induce apoptosis. So in a normal human, these mechanisms are generally enough to limit and kill infections and keep the host healthy. However, in HIV, which takes place in 3 stages, this isn't enough. So initially, HIV enters the bloodstream through an open wound, and the virus adheres to the cell membranes of the helper T cells and macrophages. They then colonize them and release their contents into the cell. Once inside, the virus utilizes reverse transcriptase to make its viral RNA into DNA, which is then inserted into the host genome. The infected cell will then begin to create new viral proteins. This will continue until the cell runs out of nutrients and supplies, and then eventually the cell will lyse, releasing thousands of new viruses into the bloodstream, allowing the whole process to take place again. So as the number of viruses skyrockets, helper T cells and macrophages begin to die off as more and more viruses infect and override their DNA to create new viral proteins. At this point, this is called phase 1 of the infection. I'll change the slide so you can see the graph. It's important to note that the efforts put in by your body at this point help combat the viral growth and offer a small buffer period where the body tries its best to restore its supply of helper T cells and macrophages. And this initially lasts a few weeks. During the second phase of infection, the immune system continues to fight as best as it can, but the level of viruses in the blood starts to grow exponentially. Steadily, the helper T cells and macrophages are picked off and killed by the cytotoxic T cells. And then finally, we're going to get into stage 3 of the infection. So it's important to note that in a healthy individual, we have approximately 1,000 helper T cells per microliter of blood. But at this stage, which is also called AIDS, the infected individual will have only 200 cells per microliter of blood, 200 helper T cells per microliter of blood, leaving the host immune system virtually non-existent and non-functioning. Okay, now we're going to move on. We're going to talk about how HIV is so successful at what it does. And so it's so successful through 3 main principles, and that's its rapid evolution, its co-infection, and its organ differentiation. Starting off, the rapid evolution happens so quick. So compared to our mammalian DNA, the evolution of HIV occurs 1 million times faster, which allows it to have short generation times, which allows it to accumulate more and more advantageous mutations. In addition, however, HIV has a reverse transcriptase that is very error prone. And this means that it tends to make errors that allow almost every possible point mutation within the genome to occur, which allows the HIV ample opportunities to gain advantageous mutations that will help it adapt, evolve, and thrive within the host. Moving on to co-infection, researchers initially thought that one viral particle, would infect each cell. However, they found that now 3 to 4 viral particles inhabit each host cell. This means that these are able to share their genetic information and enter the most advantageous mutations into the host genome to make more effective and more advantageous viruses that can then be spread and replicated. And then finally, the last part is the ability for HIV to differentiate. So they found that there were two different types of strains of HIV that differentiated within the body. There's the lymphocyte and the macrophage. So lymphocyte tropic G strains of HIV infect the lymph nodes, blood, bone marrow, and spleen, while macrophage tropic strains infect the colon, testicles, and the brain. So to test this theory, to make sure that they weren't jumping to conclusions, they tested the codons found in each organ, and they found that the codons that were under more positive selection and compartmentalized were 48.9% more frequent in their prospective organs than they were in other sites. And so this pretty much, one of the reasons I think this happens is because some organs are more susceptible to antiretroviral drugs, which allows for different environments that the HIV virus has to adapt to. Okay, so I know I'm almost over time, and I just wanted to thank you guys for listening. I know it's a lot of information, and I went through it really quickly, but I really love this subject, and if you have any questions, please let me know. I'd be happy to help.